how do we determine pKa's? Right, I told you, as you know, pKa is a fundamental property of the chemical nature of the acid or the base. But how do we determine it, right? I mean, you can look it up in tables. I know, it's the modern world. You can look everything up on the internet and find it. But where do those numbers come from? And the way that they're determined experimentally is by a procedure known as titration that you probably all did in high school chemistry. And what titration involves is the addition. It's an analytic method that's used to determine the amount of acid in solution. Let me give you an exact definition. Titration. It's an analytic method used to determine the amount of acid in a solution. And the idea is that we add in, we have a solution, we want to figure out how much acid is in it, and so we keep titrating in larger and larger and larger amounts of a base. Okay, and so let's figure out experimentally where that pKa for acetic acid came from. And what we're going to do, as I said, is we're going to titrate in some base, and we're going to keep measuring what happens to the pH as we do that. And we're going to make a plot of the pH as a function of the amount of base that we add. Okay, so we're going to plot the pH versus the amount of base. And in this case, we're going to use hydroxide anion that we add. And let's consider, for example, 100 mils of 1 molar acetic acid. Okay. So we have a total here of 0.1 moles. Okay. And we're going to see what happens. Now we have two reactions that we have to keep track of here. Okay. The first reaction is the one you know about. We have this acid that we're going to dissociate into its conjugate base, the acetate anion, plus a proton. Okay? That's our first reaction. And the second reaction, of course, is just the ion product of water. Hydroxide anion plus a proton really goes in this direction to give us H2O. And what's happening here? Well, what's happening here, of course, as you recognize, is that as I begin to add in increasing amounts of base, as I start to add more and more of this, I'm going to drive the reaction this way. And in the process, because these two reactions are occurring in solution, I'm going to force more of the acid to dissociate into the conjugate base, and so that I can consume these protons with the hydroxide. And that part makes sense? That's just Le Chatelier's principle, right? The more base I add, the more I dissociate the acetic acid into the acetate anion. Okay? And so what I want to plot now on two boards, you do it on these boards, is I'm going to plot here relative abundance of something. %, 100%, 0%. This is the abundance. Okay? And on this axis, 0, 1, 0.5. This is what we're going to call, on this axis, the equivalence of base. And by equivalence, I mean the amount of base that's equivalent to the total amount of acid I have in the solution. Okay? Now, clearly, you would agree. I'm going to use two different colors here. Here's my red. There it is. Okay. You would agree that when I start, this purple color will be the undissociated acid. And this red color will be the base, the acetate anion. And you would agree that when I start, very little of the acetic acid is dissociated. But once I add an equivalent amount of base, right, then I'm going to have converted all of the acid into its conjugate base. And so if I draw a line that represents this, this is what I get. Conversely, you would agree that at the beginning, right, at the very beginning, I have almost no acetate anion because acetic acid we've gone through. It's a terrible acid. It's a really weak acid. Okay? And at some point here, I'm going to have, once I add enough base, a complete equivalent to all the acid, I've basically consumed all the acid, and now everything I have left must be the acetate anion. And so, if I do this correctly, I have a nice curve that looks like that. And I'd like you to notice, obviously, as you can guess, that these lines should cross. I should have the equal amount right here at half of an equivalent. If we start off with a solution of acetic acid, because you all remember, of course, that acetic acid will dissociate, not very well, but it will dissociate into the acetate anion and a proton. And I told you that the pKa for this was what? I can't hear you. 4.76. Okay. So, if we take a solution of pure acetic acid, you would agree with me if I had acetic acid, and the most common, how many people here like acetic acid? How many people like to put it on your food? That's vinegar, right? So if you take a solution of acetic acid, and you would agree with me, right, that at the beginning, most of it is in the protonated undissociated form. That's the blue curve, right? And that there's not a whole lot of the acetate anion because it's a weak acid. It doesn't dissociate a whole lot. But if I now begin to add in equivalents of base, and what do I mean by the term equivalence? I mean one equivalent is the amount of base that equals the total amount of acetic acid I had in solution. Okay? And as I begin to titrate in these equivalents of base, what's going to happen, as you know, is that the OH minus anion that I titrate in is going to bind to that proton to form water. And because this is such a strong reaction, as I titrate in more base and consume more protons, according to Le Chatelier, I'm going to take this acetic acid and force it to become acetate anion. And so as I titrate in more base, the amount of acetate anion goes up and the amount of acetic acid that's undissociated goes down. And we can simply measure the pH at each point in time. So we measure the pH. I'm going to cheat here and put the answer that I want over here. And if we do this, what you find is a curve that looks like this. So it's basically a curve with a shape like this. I'll draw my points to match my curve. Okay. And this is what I measure. This is called a titration curve. Now, what do you notice about the curve? Anybody notice anything interesting about this titration curve? Well, look what's happening around here. The curve here is pretty flat. It's not till I get over here or up here that the curve starts to really change, and you can see that in the handout, right, which you have, and in this overhead that I'm showing you. Look how flat it is. Okay? And notice that it has an inflection point. And where is that inflection point? The inflection point is at exactly the pH, right, where the acetate anion and the undissociated acid are equal. Okay? And you know that that corresponds to the pH where the base and the acid are equal. So the logarithm term is equal to what? I can't hear you. 0. So the pH, where the inflection point is, this is the pKa. And that's how I determine the pKa experimentally. I don't have to do it just for acetic acid. You have a handout that shows the other curves I can do this with. Guess I'll draw one more on there. I can do it, for example, with ammonia, NH3+. And if I do, let's use a different color. If I do this with ammonia, what I get is a curve that looks like this. Okay, so that green curve there corresponds to the reaction. NH3+ dissociates into NH2 plus a proton. And again, same deal. As I titrate in the base and suck up the proton, I dissociate the ammonium into ammonia. And I look where the inflection point is for ammonia. And that inflection point for ammonia turns out to be the pKa, which is 9.25. And I can do this for imidazole, I can do it for phosphate, I can do it for anything I want. Okay? Nothing tricky there. I'm going to do the exact same thing, but I'm going to do it this time for phosphoric acid. H3PO4. Okay? And if I plot the pH, 12, 10, 8, 6, 4, 2, 0. You'll notice that phosphoric acid has 3 protons, right? Now that means that I'm going to have to add 3 equivalents of base for every molecule of phosphate to titrate these 3 different protons. And when I do that, what I find is a curve that looks like this. I get a curve that looks like this. Clearly, it looks like there are 3 points. Sorry, I drew this wrong. Let me draw it a little bit better. That's a little better. There are 3 separate inflection points that I get on this curve. pKa1, pKa2, and pKa3. This means that this is an example of a polyprotic acid. And for phosphate, pKa1 is about 2.15, pKa2 is about 7.2, and pKa3 is about 12.4. Okay? And really what's happening is the following. I have H3PO4, which has its first pKa when I get H2PO4 plus a proton, single negative charge. And this pKa is about 2.15. And then I have a second reaction, which gives rise to a proton, and HPO4 minus 2. And this has a pKa of about 7.2. And then I have a third reaction in which I dissociate another proton. And now I have the phosphate ion alone with a charge of minus 3. And this pKa here is about 12.4. Now, is there a reason, is there something special about that pKa of 7.2? What's the normal pH of your body, of your blood? 7.4. So because that pKa is pretty close to the pH of your blood, phosphate makes a pretty good buffer. To understand that, we're going to have to talk a little bit about what we mean by a buffer. Okay. Now, what do we mean by a buffer? A buffer, by definition, is a solution that resists a change in pH when acid or base is added. Now, I'd like you to recall from the overhead that I showed you, this one, that we could add, or for that matter, subtract a whole lot of base. And look how flat this pH curve is right around the pKa. Once I get far away from the pKa, like a pH of around 3 or a pH of around 6, if I start adding or subtracting a little bit of base, look how much I change my pH. But right around the pKa, if I add or subtract some base, doesn't really make a whole heck of a lot of difference, does it? And so the principle behind a buffer is to make something that's going to work close to the pKa of the molecule that you're using as the buffer. So how do we do it? Let's make a buffer. Okay? So to make a buffer, we're going to add two things together. We mix together a weak acid and the salt of a weak acid. As an example of this, let's make a solution of 10 millimolar acetic acid. Example, 10 millimolar acetate or acetic acid buffer. Okay? Sorry, I'm going to make it 20 millimolar. Okay? So we're going to use 1 liter of H2O as our final volume, and we will dissolve in it 0.01 molar. We will make it 0.01 molar acetic acid and 0.01 molar sodium acetate. Okay? Now, when I do that, and this is an example, this is an important, right? This is an important point. This is an example of what we're going to call a closed buffer system. Well, now you know how much acid, you know exactly how many moles I have, because if it's 0.01 molar and it's in a liter, that must mean that I have 10 millimoles total of acetic acid, and I have 10 millimoles of acetate anion. Okay? Now, let's add a strong acid to this. And you know what the pH is, right? The pH for this solution that we just made up, if I make it this way, the pH is equal to the pKa 4.76 plus the logarithm of the two concentrations, 0.01 molar, divided by 0.01 molar, and that's 4.76, right? Because this is 0. Now, let's do an experiment. Let's add 1 millimole of hydrochloric acid, a strong acid, to this solution. Okay? And we're going to try two things. We're going to add it to either 1 liter of pure water or 1 liter of our 20 millimolar acetate buffer. If we add it to pure water, our starting pH was 7. If I add 1 millimole of hydrochloric acid, that's 0.001 moles, what's our pH going to be? How many people think it's going to, I'm going to give you 3 choices. It's either 9, 5, or 3. Okay. How many people think it's 5? How many people think it's 9? How many people think it's 3? Sure. The pH went from 7 to 3. So we had a change in pH of 4 units. Let's do the same thing, the exact same thing, in our buffer. Okay? Now remember, our buffer started with a pH of 4.76. Okay? Now, what happens after we add 0.001 moles of hydrochloric acid? Well, of course, it completely dissociates. It's a strong acid, right? And so what's going to happen? This is going to give rise to 0.001 moles of protons. And what are those protons going to do? Well, they're going to react very strongly with the acetate anion, aren't they, right? And as a result, what's going to happen is the acetate anion, which used to be CH3COO minus, right? That concentration used to be 0.01 moles, right? We said it was 10 millimoles, right? And this now is we're going to have to add to this 0.001 moles. Sorry. Yes. Sorry, I apologize. This acetate anion is going to be consumed by that proton. So we're going to lose 0.001 moles of that because that's going to react with this proton. So our final concentration of acetate anion is going to now be 0.009 molar. Okay. The acetic acid that we started with was 0.01 moles. Okay? And that increased now by the amount with which this proton reacted with the acetic acid. So this now gained 1 millimole to become 0.011 molar. Okay? So what's our new pH? Well, our pH is just equal to 4.76 plus the logarithm of the new concentration of acetate anion, that's 0.009, divided by the new concentration of our acetic acid, which is 0.011. And this number, if you calculate the logarithm of this works out to be 0.09. Okay? And so, sorry, it becomes minus 0.09. And so our new pH is 4.76 minus 0.09, or 4.67. So what happened? Our buffer started out at 4.76, and it ended up at 4.67. We had a change in pH of 0.09, minus 0.09. Look at the difference. In water alone, we had a change in pH of 4 pH units. In the buffer, we had a change in pH of 0.09 pH units. Okay? The buffer consumed the protons and changed its pH only a little bit. Buffers work best near their pKa's. It's why phosphate is a good intracellular buffer, because it has a pKa that's pretty close to physiological pH where we want this to work. Now, other than phosphate, though, there are not that many molecules that have a really good pKa in the range of 6 to 8, which is frequently where we, as biochemists, we want to work. And so there are a limited number of choices until in 1966, a chemist by the name of N.E. Good came up with a bunch of synthetic buffers. And they are referred to today as good buffers. Not trying to be funny, that's really what they're called, good buffers. And I bet some of you are familiar with an example of a good buffer. And the good buffer that I want to tell you about is a buffer that's typically called tris. How many people here have used tris? Of course, you've all used it. This stands for tris hydroxy methyl amino methane. Okay? And it's a buffer. It's a buffer that you guys probably use all the time. What does its structure look like? Well, it's got 3 hydroxy methyl groups, as you know from the name. Okay. And it's got an amino group here, NH3+. And when you add a proton or subtract a proton, what you end up with is this. Okay. This is the acid. This is the base. Okay? And the pKa for this is about 8.1. Okay? Now, a couple points to make. I'd like you to notice that the pKa that we're working around here corresponds to the pKa of the amino group. You're not deprotonating the alcohol because, as I hope you all know, yes, that's right. As I hope you all know, that alcohol, right, trying to deprotonate an alcohol, good luck with that. That's got a pKa 11, 12, 13. Okay? So we're titrating this amino group. The second thing is, look what the pKa is. It's 8.1. Where is this going to be a good buffer? Between what pH range? 7 to 9. But I bet you, if you look at some experiments that are happening in labs you've worked in, maybe you're even guilty of this yourself. People are doing reactions at a pH of around 6 or 6.5, yet they're using tris, which is stupid, right? Buffers work best around their pKa's. And the reason is most people that aren't biochemists can't remember the name of any other buffer besides phosphate other than tris. So I've given you, for your use later on in life, a collection of buffers, right? And I'm going to show you them here, and you've got this list that shows you all the different pKa's for buffers that you can use. Now, would you, if you went to a banquet, right, and they had prime rib and spaghetti and steak tartare and sushi, would you just like eat the crackers and cheese? No, you'd eat everything at the buffet. This is a biochemical buffet. Feel free to use any of these buffers. Choose the right buffer. After 7:05, don't be one of these people who every time you need a buffer just reaches for tris. Okay?